Assalamualaikum, my name is Gul-e-adan. I have done my MPhil from University of Agriculture, Faisalabad. My topic of presentation is about effect of ciprofloxacin and sulfamethoxam on the activity of antioxidant enzymes of Mori. Basically, my topic of presentation is about effect of antibiotics on the activity of antioxidant enzymes of fish. When we talk about antibiotics, we basically say that antibiotics are used in human and veterinary medicines to treat or prevent the bacterial diseases. But the unnecessary and overuse of antibiotic is always harmful because they are not entirely removed from the wastewater treatment plants. The wastewater from the hospitals and direct manufacturing plants contribute to the presence of antibiotics into the water bodies. They adversely affect the non-targeted species such as the aquatic life. They are also considered as an important group of environmental contaminants because they can induce the oxidative stress which affect the antioxidant and detoxification processes of the fish. These changes can compromise the fish immunity which ultimately leads to the cellular damage. Consumption of fish treated with antibiotic also causes the human health risk. There is a diverse range of antibiotic and my research is on ciprofloxacin and sulfamethoxam. Both belongs to the different groups of antibiotic. Ciprofloxacin belongs to the fluoroquinolones group, whereas sulfamethoxam belongs to the sulfonamide group. Both are commonly used. Ciprofloxacin is used against the earache, bronchitis, and sinuses problem, whereas sulfamethoxam is used against gastrointestinal and respiratory tract infections. Both has tendency to generate the oxidative stress in the fish when used in excess amounts. When we talk about antioxidants, antioxidants are the molecules that are capable of stabilizing the free radicals. Free radicals are basically the highly reactive unstable molecules that has a lone pair of an electron, and in normal body, antioxidant enzymes have capability to deactivate and stabilize the free radicals by reducing their energies. When the normal balance between the antioxidant enzymes and the free radicals is lost, there is an oxidative stress, and the oxidative stress ultimately leads to the cellular damage. As we already know that aquaculture is a major economical sector and providing a lot of job opportunities along with meeting the growing demands of food, so that aquatic pollution is a serious and growing nuisance for the aquatic life because it increases the mortality of fish. Also, I want to develop the better insight about the toxicological effect of antibiotics on the fish. The main objectives of my study was to determine the effect of ciprofloxacin and sulfamethoxam on the GST and catalase enzymes of Mori. First of all, I collected the fish samples from the Fisheries Research Farm at University of Agriculture, Faisalabad. After the collection of fish samples, I acclimatized the fish samples in glass tank for about 10 days so that they adapt into a new environment. After the acclimatization, the fish was exposed to the different concentrations of antibiotic. The fish were divided into the three groups for both antibiotics, which were T0, T1 and T2. T0 was basically the control group where no antibiotic was added. T1 was the treatment group exposed to the low concentrations of antibiotic, whereas the T2 was the treatment group which was exposed to the high concentrations of antibiotic. After the acute exposure of 96 hours, fish sampling was done. Fish sampling was done after each 24 hours and fish was dissected with the help of dissecting box and the targeted organs such as the liver and gills were extracted out. After the collection of targeted organs, the homogenization was done and the organs were subjected to the antioxidant enzyme analysis. For catalase, first of all, we prepared the buffer solutions and then note the absorbance of catalase enzyme at 240 nanometer with the help of the spectrophotometer. For catalase enzyme, we prepared the buffer substrate along with glutathione solution and note the absorbance of GST enzyme at 340 nanometers. After recording the readings, the statistical analysis were done and results were generated. According to my results, catalase and GST activities increased significantly in the liver when exposed to the different concentrations of antibiotic, while in the case of gills, CAT and GST activities decreased significantly when exposed to the different concentrations of antibiotic. Variations in the antioxidant enzymes were observed, which were in following order. The liver were more affected as compared to the gills because liver is the main organ for the detoxification. Maximum activity was observed at 96 hours and GST enzymes were more sensitive to antibiotics as compared to catalase enzymes. Exposure to a different concentration of antibiotic caused variations in the antioxidant enzymes of fish. We say that when fish was exposed to the different concentrations of antibiotic, it affects the antioxidant enzyme activity. Variations in the antioxidant enzyme activity could be considered as a reliable biomarker for the antibiotic studies. And that's it. Thank you so much.